So my name is Jonas Muhr, and I work as a professor at the Karolinska Institute in Solna. So I started off reading biology with the intention of becoming a marine biologist. As you realize, I did not succeed with that intention. And one reason for this was that I found life science courses and courses in molecular biology most interesting. So I switched field, and in 1999 I graduated with a thesis focusing on early development of the vertebrate central nervous system. I started my lab here at the Karolinska Institute 2003, and I now have a lab consisting of 4 postdocs and 4 PhD students, where half of the lab is working with cancer-related questions and the other half is working with questions related to the developing nervous system. So this talk will be focusing on the latter, more exactly how stem cells or stem cell proliferation are regulated during brain development. But before I start to describe this project, I would like to give a more general introduction to the field. It may be a bit brief, but I hope you will follow. Okay, so what is then developmental biology? One broad definition, but good definition I think, is how a new individual is formed from a fertilized egg. Important questions that often are asked are, for instance, how are differences between cells created? You all know that there are differences between a brain or a neuron and a skin cells. How can these differences be established during development? How can we explain the differences between normal and abnormal development? And how can we increase the cell number during development? Remember, an embryo we have to increase quite substantially in size before it's born, or the fetus is born. So this is not a new scientific field. Already the ancient Greek scientists and philosophers thought about these questions. For instance, the medical scientist Hippocrates, he was examining embryonic development by studying developing chick embryos. During the 17th century, there were 2 schools in this field. There was one school that believed in something called preformism, which claimed that organisms, they develop from miniature versions of themselves. So all organs and tissues existed prior to development. And these guys, they got experimental support from early microscopists, like this guy Nicholas Hartzocker from the Netherlands, who claimed to see a small tiny person inside the head of the sperms. There were other microscopists that claimed to see tiny persons inside the female egg. And they claimed that the sperms, they were just parasites on the female egg. Now we know that this is not completely true. The other school during this time believed in something called neoformism, which is more similar to what we believe today, namely that organs are formed gradually from an undifferentiated mass by a series of steps during which new parts are added. But the cells and the cell theory was first proposed, or the existence of cells and the cell theory was first proposed during the 19th century. And that theory claimed that all organs consist of cells, which is true, and that growth of organ is dependent on cell divisions. Now we know that all cells contain DNA, which contain genes that encode for protein and that regulate all cellular behaviors and also what cells gonna become, what type of cells they will become during development. So this talk will then focus on one aspect of brain development. This slide shows a section through the human brain, which you all know is a very complex organ. It contains approximately 100 billion of nerve cells, which can be compared to the number of stars in our galaxy. These neurons can be categorized into approximately 1,000 to 10,000 different subtypes. And if you think about that, each nerve cell can make up to 1,000 connections. It's easy to appreciate how complex this organ really is. So most neurons, they're generating during embryonic stages, and they're generated by stem cells. So during a period of 9 months, the brain will develop from a small rudiment into this highly sophisticated organ consisting of 100 billion of neurons. So then it's easy to understand how high demands brain development put on stem cells and on the molecular pathways that regulate stem cells. So what are stem cells? Stem cells are immature cells, and they have two important features. They can self-renew, and I should say it's a very long-lived cell type that in many organs persists throughout life. Anyway, they have two important features. One feature is that they can self-renew, and generate new stem cells. But they can also differentiate and generate so-called differentiated progeny, such as mature neurons. Depending on how many different cell types stem cells can generate, one talk about different grades of potency. So we have totipotent, we have pluripotent stem cells that can generate many, many different cell types. And we have bipotent or unipotent stem cells that can generate just a couple of different cell types. Stem cells in the brain, they're said to be multipotent, and then they can generate often a number of different neurons and glial cell types. So this slide shows a section through the embryonic mouse forebrain, where the stem cells, here shown with the white color, are located in a structure called the subventricular zone, that is next to a hollow structure called the ventricular system. As the stem cell generate neurons, they have to go through a cellular process called neurogenesis. The major cellular steps during neurogenesis are summarized in this model figure, where a self-renewing neurostem cell either can develop toward glial fate or in the presence of a neurogenic signal towards the neuronal lineage. In the presence of a neurogenic signal, the neurostem cell will form something called a neuroprogenitor cell. And the progenitor cells are, in comparison with stem cells, they're not as long-lived. So they will just live for a short period of time. And during this time, they will go through a number of rapid cell divisions before they finally exit the cell cycle and start to differentiate into neurons. So, how can this rather limited population of neurostem cells that we are born with, or that we have that make up the brain or the nervous system, how can they generate 100 billion of neurons? Of course, they have to self-renew to do this. But one should remember that each cell division is connected with the potential risk of incorporating oncogenic mutations into the genome. And as you know, oncogenic mutations can cause aberrant cell proliferation and finally cancer, which would be devastating for a long-lived cell population like stem cells. So stem cells, they have solved this problem by generating something called a transient amplifying cell type that go through that is much more short-lived and go through a number of rapid cell divisions before they finally differentiate into a mature cell type, such as neuron. So in the brain, or in the developing cortex, it will look something like this, where a stem cell here, this is the brown color here is the subventricular zone, and where the stem cell, or the radial glia cells, as they are called, divide very slowly, once in a while. They generate a so-called transient amplifying cells, or an intermediate preennate cell, that go through a number of rapid cell divisions and form mature neurons. So what this means is that one division by a stem cell will result in the generation of many neurons. So what we asked a couple of years ago was, how are stem cells regulated so they can be maintained in a slowly proliferative state? And why are these preennate cells allowed to proliferate fast? So how is this regulated? And to address this question, we have been focusing on a transcription factor called SOX2. This panel figure shows a section through the embryonic forebrain, where the expression of SOX2, here shown in red, shows that SOX2 is expressed in the majority of all stem and preennate cells in the subventricular zone. This figure also shows that SOX2 is strictly downregulated before the cells, the differentiating cells, start to upregulate neuronal markers, here shown with the green color. So what is a transcription factor? Well, it's a factor that can regulate the expression of other genes. So it can either associate with a co-activator and activate gene expression, or it can associate with the transcription repressor and repress gene expression. So one gene can regulate many other genes. So why SOX2? And when we are studying stem cell proliferation? Well, there are some previous data suggesting that SOX2 actually can be involved in regulating proliferation. For instance, if one overexpresses SOX2, that leads to a reduction of the proliferation of cells kept in vitro, in cell flasks. That has been shown before. If you do the opposite and reduce the level of SOX2 expression, that can cause quiescent. Quiescent is cells that do not proliferate at the moment. Quiescent Müller cell preennators in the adult mouse retina to start to proliferate. So that's the opposite scenario from this one. And what we and others have found that in the developing cortex, even though SOX2 are expressed in, or is expressed in all cells in the subventricular zone, it's expressed at high levels in the so-called stem cells and at lower levels in the cells that we call preennate cells. For instance, the level of SOX2 can be examined in vivo with the help of antibodies and immunohistochemistry. This panel of figures show a section through a cortical half, where the expression of SOX2 here shown in is here shown with the green, and the expression of the thymidine analog BRDU, which in here only label proliferative active cell, or cells that are proliferative active, are shown in red. If we now categorize the cells depending on the level of SOX2 expression in 10 different bins, we see that the red cells, or the cells that have incorporated BRDU, they end up in the bin where we have cells expressing low levels of SOX2, whereas the cells that express high levels of SOX2 are much less proliferative active. So there's a negative correlation between SOX2 levels and proliferation. So could this mean that SOX2 actually can be a determinant regulating proliferation in the cortex, depending on its expression level? And to address this question, we have been using a system called mouse embryo electroporation. It's very neat because we can then very easily increase or decrease the level of SOX2 expression in the subventricular zone. The experimental setup is that we inject vectors overexpressing SOX2 or vectors expressing something called shRNA that knock down SOX2 expression. We inject these vectors into the ventricles of the embryos, meanwhile they're still in the uterus. Thereafter we apply current over the forebrain, and that will lead to a transfection of these vectors in the cells that are within the subventricular zone. And this is how the misexpression can look like 48 hours after electroporation. So this is an embryonic cortical half. So what happens then if we now overexpress SOX2? Well, what we found, if we now overexpress SOX2 GFP, so this is a construct where we link SOX2 to GFP. So one should focus on the green cells. We see that the cells quite rapidly respond by decreasing their proliferation rate. If we now decrease the level of SOX2 expression with this construct called shRNA, we instead nearly double the amount of proliferative cells. So this means that depending on the level of SOX2 expression, we can actually modulate proliferation either down or up. So how can now SOX2 achieve this regulatory function? So how is it done molecularly? And to address this question, we have done a number of different experiments. We have, so this is a cortical half again. What we have done, we have used something called chromatin immune precipitation, or ChIP-seq experiments, which is a way of determining where in the genome or to which genes SOX2 is binding in cortical stem and prehended cells. And to understand which of these genes to which SOX2 binds that are regulated when we alter the level of SOX2 expression. We have also misexpressed SOX2 in cortex and used shRNA to reduce the level of SOX2. Whereafter we have checked with something called RNA-seq experiments to see which genes respond when we overexpress or lower the level of SOX2 expression. So these experiments went well. So we found thousands of genes that are bound by SOX2 in the cortex. And we found several hundred genes that are upregulated when we overexpress SOX2. And we found several hundred genes that are downregulated when we overexpress SOX2. And similar with the shRNA, when we reduce the level of SOX2, we found several hundred genes that are both up and downregulated when lowering SOX2 levels. So which of these genes are then interesting for us? That's not super easy to answer. But we have used something called gene ontology analysis, which is a way of connecting a cellular process to a gene set. And what we found with this analysis was that one group of genes stood out here because they got high enrichment score for the process called, or the cellular process called, neural precursor cell proliferation, which was something we were studying. And this set of genes were the genes that were downregulated by SOX2 overexpression and or upregulated when we knocked down SOX2 levels. So this suggested to us, okay, maybe SOX2 is actually decreasing the expression or reducing the expression of genes that are in turn promoting proliferation. So we wanted to test this. So we took a subset of these genes that were actually both downregulated by SOX2 expression and upregulated by SOX2 knockdown. And we tested them all in the embryonic cortex and checked whether they could promote proliferation. And 4 out of these 9 genes promoted proliferation when they were electroporated into the cortex. But only 1 of these 4 genes, namely the gene that encodes for the cell cycle regulator cyclin D1, could fully retain its ability to promote proliferation, even when it was co-expressed with high levels of SOX2. And this is something that is expected from a gene that is acting, that promote proliferation downstream of SOX2 repression. So these data suggested to us that that SOX2 reduce proliferation in stem cell by repressing this cell cycle regulator cyclin D1. So our data suggests that cyclin D1 is expressed at low levels in stem cells due to the high level of SOX2 expression in these cells, and that the level that leads in turn to their slow proliferation, and that the level of cyclin D1 is allowed to increase in transient amplifying progenitors due to the decrease in SOX2 expression, and that will in turn lead to enhanced proliferation in these cells. So how can the different levels of SOX2 now regulate the expression of cyclin D1? To address this question, we have been focusing on the promoter region of cyclin D1, partly because it has been shown. I should mention that the gene that encodes for the protein cyclin D1 is called CCND1. That's why you see this label there. Anyway, so why we focused on the cyclin D1 promoter is because it has been shown to regulate regulate the expression of cyclin D1, and that our ChIP-seq experiment demonstrated that this region is one of the regions in the genome that is most massively bound by SOX2, here represented with the massive peak. So how do we determine, how can we examine how a promoter region is regulated? Well, what you normally do is that you isolate it, and then you can clone it in front of, in this case, or a reporter gene, in this case, luciferase. This luciferase is not a gene that is normally expressed in vertebrae or in the cell types that we work with. What we did with this cyclin D1 reporter vectors was to transfect it into cells with or without vectors overexpressing SOX2, or with vectors that express a dominant negative version of SOX2 that decrease SOX2 activity. And what we found was that the promoter region of cyclin D1 responded as the endogenous cyclin D1 and was decreased by SOX2 overexpression, and increased when we lowered SOX2 activity using this dominant negative version of SOX2. As endogenous cyclin D1, this reporter responded gradually or responded in a dose-dependent manner to increasing levels of SOX2. So how can then SOX2 regulate? How can SOX2 achieve its regulatory function on this on the cyclin D1 promoter? Well, if we now look more carefully into this promoter region, we see it contains many SOX sites or motifs, 9 SOX motifs that we denoted SM1 to SM9, whereas the motifs here labeled with a red color contain SOX or consensus SOX motifs that are bound by SOX2 with high affinity. whereas the motifs labeled with the green color, they contain more degenerated versions of the SOX binding sites. And to these sites, the SOX2 protein bind with less affinity. So we have many sites, but with different affinities. And what we found interesting was that if we now start to mutate these different sites, destroy the SOX sites, we found if we now destroy the consensus site, or the site where SOX2 bonds with high affinity, not very much is happening in terms of repression. It can SOX can still repress the promoter without these sites. So it should be compared to this wild type bar. But as soon as we start to mutate the off-consensus motif, the motifs with less affinity, we start to lose the repressive capacity of SOX2. And if you mutate all of these off-consensus site, the green motifs, we completely lose the ability, or SOX2 completely lose its ability to repress this reporter. So these results have led to a model where SOX2 in stem cells is expressed at high levels. That enables SOX2 to bind both to consensus sites, high affinity sites, and off-consensus site, low affinity site in the cyclin D1 promoter. This will in turn lead to efficient reduction of cyclin D1 expression and will maintain these cells in a slow proliferative state. As the stem cells start to differentiate into progenitors, or transient amplifying cells, or in this case intermediate progenitor cells as they are called in the cortex, the level of SOX2 is dropped. And that will lead to that only the high affinity sites will be bound in the cyclin D1 promoter. And that will also result in a less efficient reduction of the cyclin D1 expression. So that the repression of cyclin D1 will be relieved, and as a consequence, we will have more cyclin D1 protein, and the cells will proliferate with a faster rate. So this result represents a nice example where actually the presence of high affinity and low affinity binding motif allow a transcription factor to act in a dose-dependent manner, and thereby regulate gene transcription differently, dependent on it if it's expressed at high levels or low levels. So I will finish there by acknowledging the guys who have done the work. They're all here. This is on a camel ride in Marrakesh. And they're very shy, as you see, so I only show their shadows. But I should especially point out Daniel Hegge, that did most of the work presented here today. And I'm here in the front. Okay, thank you.